Welcome to AMiNDR, a podcast where we summarize the latest publications on neurodegenerative disease research so that you can stay up to date with the newest findings. Every month, our team of scientists will sort and organize the titles into themes and present shortened versions of the abstracts. We'll make sure to mention the title, the journal, the first author, and the last author for each publication. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast helpful.
Welcome back to another episode of AMiNDR. This is Anusha, and I'll be your host for this episode. We'll be covering all of the papers that discuss cognitive and behavioral changes associated with AD in terms of disease mechanisms or pathology that were published in August of 2020. We've actually got quite a bit to work with here, 18 papers. So let's get started. Oh, and before I do, I just want to remind you that in the episode notes is a link to the bibliography, so you don't have to memorize the titles of these papers you find interesting, unless, of course, you want to. The first paper we'll be discussing comes from the Journal of Clinical and Experimental Neuropsychology. This was written by first and last authors Sandoz and Fossard and is called Referential Adjustment During Discourse Production in Alzheimer's Disease. Authors here looked at changes to vocabulary that are associated with AD. Interestingly, individuals with AD tend to use more pronouns than their healthy controls when speaking of other entities. This is called referential behavior, and it's actually inversely correlated with cognitive function, including working memory. In this study, authors looked at 3 stages of discourse: the introduction of a referent, maintaining the referent in focus, and the shift in focus from one referent to another. 21 people with AD and 20 healthy adults participated in a storytelling task. Through this, they found that individuals with AD during the introduction and shift stages produced significantly fewer referential expressions. They suggest that different cognitive competencies, including executive function, are important in selecting referential expressions. Moving on, we have semantic loss marks early Alzheimer's disease-related neurodegeneration in older adults without dementia. This was published in Alzheimer's and Dementia Amst by Vonk, Dufoil, and others. Authors here assessed changes to verbal fluency in 2,261 non-demented individuals and followed them up for 4 years as they began to fall into cohorts of those with no mild cognitive impairment, or MCI, those with amnestic MCI, non-amnestic MCI, or incident dementia. They also related these assessments to MRI and PET imaging during a 2-year follow-up. They found that in carriers of ApoE4, those individuals are at a higher risk for AD, and in other high-risk groups, semantic fluency declined the fastest while letter fluency did not, with the exception of individuals with incident dementia. Lower semantic fluency was also correlated to an increase in the white matter hypertensities, lower hippocampal volume, more cortical thinning, and reduced dye uptake in PET imaging of brain areas including the entorhinal cortex, precuneus posterior cingulate area, and others. Neurodegeneration of the whole brain was associated with decline in both semantic and letter fluency, and AD-associated regional degradation was associated with more stark decline in semantic but not letter fluency. Up next, we have a paper from authors Noclave, Soininen, and others who published this paper in Cognitive Science. This is called Production of Inflected Novel Words in Older Adults with and Without Dementia. Researchers wanted to look at how older adults pronounce or inflect new words, as children use words they already know to guide this decision. They had participants perform a simple speech elicitation task. By the way, this paper came out in Finland, so we're actually focusing on the Finnish language. In the task, they had pseudo-Finnish words mixed in with actual Finnish words, and cohorts of those with MCI, AD, and healthy adults perform this task. In both cohorts of healthy individuals and those with AD, resemblance of novel words to known ones, as well as bigram frequency, which are 2-word chunks, significantly influenced the choice of inflection for these novel words. They suggest that this therefore supports a single root model through phonological analogy instead of the commonly proposed dual route model. Next, in PLOS 1 by Stefaner, Hill, and one other author, neural capacity limits on the responses to memory interference during working memory in young and old adults. The optimal way for the brain to perform a task is to recruit as few neurons as possible. As we age, the recruitment of extra brain regions to complete a task is actually observed, so we're not as efficient anymore. During verbal working memory retention in older adults, additional recruitment of the parahippocampal brain regions during interference is observed than in the absence of interference. They hypothesize that intrinsic noise produces internal interference with age, and that this requires the recruitment of these additional brain regions to combat that. They used a verbal delayed item recognition task with different levels of interference during the maintenance of information acquired through working memory. There was an age-related trend, but no significant interaction effect between age groups. In more specific brain regions, they did see significant interaction between interference and memory load. But check out the paper for more details. In the Journal of Clinical and Experimental Neuropsychology, we find our next paper. This is authored by Roquette, Lemaire, and one other. Alzheimer's disease disrupts domain-specific and domain-general processes in numerosity estimation. In this paper, they sought to quantify numerosity estimation abilities in AD. This could mean, for example, guessing the number of items in a collection. Healthy older adults were compared to AD patients in the DOT comparison task and Simon task to assess number estimation abilities. I won't go into detail about these findings, but I will tell you what they concluded from it. They found that numerosity abilities decline in AD, both in number processing and general inhibition. So it's not just brain region specific. The next two papers are going to be looking at a genetic model of AD, the 5X FAD mouse. I just wanted to switch things up a little, so we're deviating from cognition to olfaction here. Alterations in odor hedonics in the 5X FAD Alzheimer's disease mouse model and the influence of sex is the title of this paper. This was published in Behavioral Neuroscience and was written by first and last authors Roberts and Wesson. Researchers used a genetic AD model, the 5X FAD mouse, to test if olfactory impairments, which they have previously observed in other models, are observed with this particular genetic mutation. They found that 6-month-old 5X FAD mice investigated odors longer than controls did, and more so in females. They also saw heightened odor preference in these female mice, suggesting aberrant hedonics. They suggest that this is indicative of age and sex-specific changes to hedonic behavior in these mice. The next paper is also looking at the 5X FAD mouse, but in another system, vision. It's called Retinal Function and Structural Changes in the 5X FAD mouse model of Alzheimer's disease. This was published in Frontiers in Neuroscience and comes to us from the keyboards of Lim, Nguyen, and others. If you are unfamiliar, 5X FAD mice have a genetic mutation that leads to A-beta deposition over time and has been shown to exhibit dementia-like symptoms. Using optical coherence tomography, they analyzed the retinal nerve fibers in these 5X FAD mice over time and found that after 6 months, the mutated mice had thinner retinal nerve fibers than their wild-type littermates. Weirdly enough, the inner plexiform layer at 6 months was thicker in the 5X FAD mice. Function of the retinal ganglion cells was reduced in the mutant mice and spread to the outer retina at 17 months of age, but this was not observed in younger mice. They actually found insoluble A-beta deposits in the retina at all ages they tested in these mutant mice. So taken together, it means that the inner retina seems to be more sensitive to the functional consequences of the mutation, and therefore A-beta accumulation. Let's climb back up the evolutionary ladder and shift our focus back onto humans. This next paper is called Limbic Predominant Age-Related TDP-43 Encephalopathy, or LATE for short, ADNC Pathology and Cognitive Decline in Aging. This was published in Neurology by first and last authors Kapasi and Schneider. Authors here looked at the late neuropathological changes called late-NC and compared them to the AD-NC group and those who had both, so mixed AD and late-NC group. And they also compared this to those who served as healthy controls. They used performance on annual cognitive testing alongside systematic neuropathological examination at autopsy. They related this through mixed effect models to identify rates of decline in global cognition and five specific cognitive domains and correlated these to age. The pure late-NC group showed faster decline in global cognition and episodic memory when compared to controls, but slower decline when compared to the AD-NC group. Mixed AD-NC and late-NC demonstrated the fastest decline, with faster decline occurring before 90 years and slower decline occurring after 90 years old. Now we're moving on to the last paper we'll be covering before the halfway mark. And this was authored by Kilecioglu and Raman, as well as another Raman. As always, forgive my pronunciation. Published in Applied Neuropsychology Adult, this is called The Influence of Age of Acquisition on Recall and Recognition in Alzheimer's Patients and Healthy Aging Cohorts, in Turkish. Age of acquisition, or AOA, may affect things like word recall and recognition, and researchers here wanted to determine just that in 30 Alzheimer's patients and 28 healthy age-matched controls. Participants were given 20 words that varied critically on AOA and were also tested using the WAIS-R and mini mental state examinations. They found that AD patients demonstrated worse recall and recognition, and that early acquired words were better retained than late acquired words. They go into detail in this paper as to what this means for current theories of the impact of AOA on loss of memory, so be sure to check out the paper for the significance of their findings. I'll take a moment now to remind you to check out the episode notes for a link to the bibliography, which we've conveniently timestamped, so you know exactly which entry to look for. Alright, I'll catch you on the flip side. Welcome back, everyone. Let's not waste any time here. The next paper we'll be looking at comes out of the Journal of the American Geriatric Society. It's called Neuropsychiatric Symptoms and Functional Decline in Alzheimer's Disease and Lewy Body Dementia. First and last authors, Borda, Vikmo, and others aim to better understand how neuropsychiatric symptoms associate with functional decline in individuals recently diagnosed with AD or Lewy body dementia. They were followed up for 5 years, and by the way, this study looked at patients out of Western Norway. On the Norwegian Neuropsychiatric Inventory, or NPI, a linear mixed effects model was used to analyze the data. They found that NPI longitudinal scores demonstrated functional decline in both cohorts, and that the baseline total NPI score was able to predict the decline in Alzheimer's disease. Let's step back into the visual system for the next two papers. The Journal of Alzheimer's Disease is where we'll find this next one, which is titled Alzheimer's Disease Progressively Alters the Face-Evoked Visual Processing Network, by Huang, Zhu, and others. In this paper, face-evoked visual processing was tested in 8 patients with severe AD, 11 with mild to moderate AD, and 26 controls. In healthy subjects, the visual processing network started in the V1 brain region and ended in the fusiform gyrus. This is in contrast to the AD patients who showed disrupted networks that were dependent on severity of the disease. The milder form reduced and disrupted higher-order visual association areas, whereas severe cases showed virtually no network at all in these areas. Next, we have Egocentric and Allocentric spatial cognition in amnestic mild cognitive impairment and Alzheimer's disease. This paper was published in European Neurology by first and last authors Hashimoto and Komori. I should add that there are a fair amount of 3-author papers, including this one, and I feel kind of bad for leaving out that last author, or I guess the middle author, but it's okay. Maybe it's more incentive for you to check out these papers. Researchers here wanted to further explore the nature of topographical disorientation, which is an early symptom of AD. They did this by looking at egocentric and allocentric spatial cognition in participants with amnestic MCI and early AD, as well as in healthy volunteers. By my understanding, they used a card placement test and asked participants to place 3 cards onto grids that surrounded the individual before and after asking the individual to turn around. Then they were asked to recreate the array of the 3 cards. I tried to look up further details, but unfortunately I didn't have full access to the paper, so if you're interested, try your institution's VPN or contact the authors directly to get access to their paper. They found that amnestic MCI patients performed much poorer in allocentric placement, and AD patients performed equally poorly on both forms of spatial memory. The AD patients also demonstrated more errors in card placement in both ego and allocentric tests, suggesting that egocentric spatial cognition can be used to distinguish AD from amnestic MCI patients. Up next, we have the Journal of International Neuropsychology, from which we'll be looking at a paper titled Emotion Recognition and Traffic-Related Risk-Taking Behavior in Patients with Neurodegenerative Diseases. This is by first author Vandenberg and last author Huitema. Authors here sought to assess whether impaired emotion recognition, which is seen in several neurodegenerative diseases, is correlated to risky decisions, such as those made in traffic. 51 patients with various neurodegenerative diseases were asked to perform a facial expression of emotions, stimuli, and test, or feast task. This was paired with a driving simulator and an action selection test and was compared to age-matched healthy controls. They found that patients were significantly worse at recognizing anger, disgust, fear, and sadness, and took significantly more risks when driving, or in the driving simulation, I guess, and action selection tests. Poor recognition of fear was the only emotion related to dangerous driving. So, authors suggest that when assessing if patients are fit to drive, measuring emotion recognition might be an additional tool that can be used. This next paper is a single-author paper from Lee and was published in Current Alzheimer Research. It's called Impaired Cognitive Function is Associated with Motor Function in Activities of Daily Living in Mild to Moderate Alzheimer's Dementia. Lee, in a South Korean setting, measured the relationship between cognition, motor function, and activities of daily living in Alzheimer's disease dementia patients. These individuals were tested using the MMSE, so the Mini Mental State Examinations, clinical dementia ratings, and global deterioration scales. Motor function was assessed using a 10-meter walking test, Berg Balance Scale, and timed up-and-go tests. Activities of daily living were assessed using the modified Barthel Index and Katz Index. Together, the data show cognitive function by Mini Mental State Examination was correlated with Berg Balance Scale and the modified Barthel Index and Katz Index. The Mini Mental State Examination scores, which measured cognition, can also explain the variability in the other 3 scores that I just mentioned. This suggests that decreased cognition affects motor function and activities of daily living. Perhaps interventions such as physical exercise can help address all 3 areas. Next, out of Dementia and Geriatric Cognitive Disorders, is a paper called Structural Brain Network Correlated with Reading Impairment in Alzheimer's Disease. The first author is Oda, and the last author is Arai. Researchers aim to identify the relationship between language functions measured by the standard language test for aphasia and the brain network connectivity of Japanese AD patients. They had 22 patients who had an MRI scan and the language test I just told you about, the SLTA, from which researchers determined brain network connectivity. A significant negative correlation between reading out loud in kanji words and the clustering coefficient in several brain regions were observed. Significant negative correlations between the clustering coefficient in the left prefrontal region and the auditory comprehension of words was also found. You can visit the paper for more specifics about brain regions affected. All right, now the home stretch, just 3 more papers to go. The first was published in Otology and Neurotology by Aylward, Gergel, and others. It's called Changes in the Auditory Association Cortex in Dementing Illness. Here, authors wanted to see how well correlated the degree of cognitive impairment was to gray matter density changes, specifically in the auditory cortex. They used MRI data from 663 patients at a cognitive disorders clinic and found that the ratio of auditory association cortex to whole brain in terms of gray matter densities was significantly different in patients with a mini mental state examination or MMSE score of less than 25. This, along with no change in primary auditory cortex to whole brain ratio, suggests that patients with more severe impairment may have problems processing but not perceiving sounds. Hamilton, Thomas, and others authored this next paper in the American Journal of Geriatric Psychiatry. This is Cognitive Decline in Mild Cognitive Impairment with Lewy Bodies or Alzheimer's Disease, a prospective cohort study. Here, authors used a longitudinal prospective study to track patients diagnosed with dementia with Lewy bodies, or AD, after 5 years, and these patients came out of the UK. An MCI cohort was separated into MCI due to AD or probable MCI with Lewy bodies, and these individuals were tested in a battery of clinical assessments. Researchers found that probable MCI with Lewy bodies showed less impaired recognition memory than the AD group and greater impairment in verbal fluency and perception of line orientation, which was further impaired as time went on in the Lewy body group. It seems that visual spatial function is compromised to a greater extent with dementia with Lewy bodies as compared to AD. This is it. The final paper. All right, this lucky number was published in Current Alzheimer Research and is called Self-Defining Memories and Their Contribution to the Sense of Self in Alzheimer's Disease. There are only 2 authors here, Hadj and Alain. Memories of very important personal events are referred to as self-defining memories, and this differs from autobiographical memory, which is more for personal information. Self-defining memories were evaluated in AD patients and in controls for certain characteristics. Production of positive or integrated self-defining memories were not different in AD participants, nor was there a difference in the rating of centrality and its contribution to self-continuity. Ultimately, although AD patients produced fewer specific self-defining memories, they are similar to controls in emotional valence, integration, as well as centrality and self-continuity of these memories. And we're done here. Before I turn it over to Sarah, I'll just take a moment to mention that she actually met with the gentleman at the Brain Buzz podcast to do a crossover episode. So be sure to watch out for that, which is set to come out in October of 2020. Thank you so much for listening to this episode, and I hope you have a wonderful rest of your day, however that unfolds. Bye-bye.
That's it for this episode. A huge thank you to the team that is working on sorting, summarizing, and scripting these abstracts, as well as the operations behind AMiNDR. The music is from Journey of a Neurotransmitter by Anusha Kamesh, musician and fellow scientist, and now a member of the AMiNDR team. You can find the original piece and her other music on SoundCloud under Anusha Kamesh or on her YouTube channel, AK Music. Interested in joining the team? Give us a shout! We can always use help with content development, podcast editing, advertising, and you can be part of a new and exciting venture. Reach us by email at aminderpodcast@gmail.com or follow us on Twitter. Oh, we're also on Facebook now. Don't forget to subscribe to our mailing list if you want access to the bibliography for each of our episodes. The references come with timestamps. Timestamps.
so you can more easily locate the paper that caught your interest. Check our notes below for details on how to sign up. And very close to this, you'll also find a link to our feedback survey. Because, yeah, your feedback matters to us, so please, pretty please, let us know how we can make this podcast a better tool for you. And last but not least, thank you for tuning in with us. And on this note, we hope you found our podcast useful and accessible. Until next time.